Okay, it's my great pleasure to introduce Dr. Kevin Devine, who's going to talk to us about DNA in space.
Thank you. Thank you. Thank you. Okay. Thank you for coming. My title of my talk is An Astronaut in Structure Space. By structure space, I mean molecular structure space. The great thing about being a chemist is we can make analogues of naturally occurring biological molecules and test their behaviors in the lab to try and see could different types of biopolymer in particular, in my case particularly nucleic acids, could they show the same sort of behaviors and act as genetic molecules with the same fidelity as do the naturally occurring nucleic acids, DNA and RNA. And biopolymers possess key intrinsic features. Nucleic acids, they're heterocyclic aromatic bases appended to a polyanionic sugar phosphate backbone. The sugar, phosphate, and coming off the sugars are these sort of like planar ring structures known as heteroaromatic bases. Now proteins, they're quite different. They are amino acids linked by polyamide bonds. The polyamide known as peptide bonds. That gives you your proteins. Best known group of proteins are the enzymes which catalyze metabolism in all living organisms here on planet Earth. Polysaccharides, these are carbohydrates linked by acetyl ether groups. And if you're a chemist like myself, you look at these structural features and you ask the great question, why? Why have these different classes of biopolymers been constructed by nature in this way? And nucleic acids is the focus of my talk. I'm going to focus entirely on this group. They're biopolymers built from nucleotides. Now, a nucleotide itself consists of three components: a heterocyclic base known as a nucleobase, a sugar, and phosphate. Now, the sugar is either ribose, in the case of RNA, while in DNA, it's deoxyribose, 2' deoxy D-ribose, and by that we mean at position 2', which you'll see in a minute, the next slide, it lacks an OH group, hence deoxy. The term base for the heteroaromatic component is a bit of a misnomer. As a chemist, when a chemist thinks of a base, thinks of something that's highly alkaline, strongly alkaline, such as sodium hydroxide. Well, these aromatic systems are not basic to any real degree. But it was a time when anything containing nitrogen was considered basic, and unfortunately it's a sort of cultural term that has lived on to this day. We continue. Nucleobases, and there are two distinct types. As purines, these are 9-membered aromatic rings, adenine and guanine, or A and G, if you're familiar with the sequences of DNA, A and G. And pyrimidines, these are the smaller 6-membered rings, and these give rise to the bases in DNA, cytosine and thymine, and uracil in RNA. RNA, the base adenine, cytosine, guanine, and uracil. And DNA, thymine replaces uracil. So we're going to look at the structures of these components. Next slide. And there they are. Adenine, guanine, cytosine, uracil, and thymine. Thymine is like a 5-methyl uracil. And these are the two sugars. There's D-ribose and D2 deoxyribose. As you can see, this lacks an OH group in the 2' position. And here's the numbering system. When you've got two components in a molecular structure, you want to you must distinguish between which one you're numbering. So for the nucleobases, you have the ordinary numbers from 1, 2, 3, 4, 5. The sugars are given primes in front of their numbers. So there's the 1', and the 1', the two 1 and 1' are joined together. They're adjacent to each other. As you can see there, when X is H, we have a deoxyribose. That occurs in DNA. And the nucleosides, here they are, adenosine, guanosine, cytidine, and uridine. These are the nucleoside components of RNA. And for DNA, we have 2' deoxyadenosine, 2-deoxyguanosine, 2-deoxycytidine, and thymidine. As thymidine doesn't exist in RNA, it doesn't have the deoxy in front of it. And oligo or polynucleotides, as you can see, these are linked via phosphate. We've got phosphates linking the two, and they're linked 3' to 5' in both DNA and RNA. And the sequence goes from the 5' end to the 3' end. So when you see in a textbook a sequence written down, the left-hand end is going to be 5', right is going to be the 3' end. Now, duplex oligonucleotides. This is where the genetic information is stored in the sequence. And it's a base pair in duplex. That's the essential component of the duplex itself. There's a base pair. And there are 2 complementary strands held by base pairing on opposite sides. And the base pairs use 2 complementary principles: size complementarity, where small pyrimidine with a large purine, and hydrogen bonding complementarity, which we're going to see. Next slide. And the elucidation of the double helix is just over now. That was just over 60 years ago. And it was this photograph of DNA crystals by X-ray crystallography. It was taken by the late Rosalind Franklin in the laboratory of Maurice Wilkins at King's College on the Strand. And this is very strange. If you look at it, this is very strange, because now to decipher what structure gives rise to that essentially 2-dimensional photograph. Well, it was a couple of scientists. One was the physics-trained Francis Crick and James Watson were intrigued by this and started building models to try and explain it. And they found the best model, 3-dimensional model, that could give rise to that X pattern in essentially a 2-dimensional photograph, was a double helix. And they were set about building models to try and explain this. And to date, this is the familiar double helix, a sort of right-handed helix with 2 distinct grooves, a major groove and a minor groove. And the Watson-Crick hydrogen bonding base pairs, there's the CG base pair. Remember, C, the pyrimidine base, a small 6-membered ring, makes hydrogen bonded contacts with a large purine, in this case guanine. And its PYD stands for hydrogen bond donor, which is shown in red. And the hydrogen bond A stands for hydrogen bond acceptors, which are shown in blue. So the NH groups are hydrogen bond donors, and they interact with the lone pairs on either oxygen or nitrogen atoms in the complement base. And the TA base pair, small thymine or uracil in RNA, binds with adenine, the purine, in both DNA and RNA. And you notice that the CG base pair is held together with 3 Watson-Crick hydrogen bonds, the TA base pair with just 2. And here's a duplex between 2 complementary nucleic acid strands. And you notice that one strand runs 5', 3', and its complement run in the opposite direction. So those are 2 anti-parallel complementary strands. And you can see G binds with C, T binds with A. Now, in addition to Watson-Crick hydrogen bonding, there are so-called non-Watson-Crick H-bond pairings. If you shift the G base down slightly, it forms a so-called wobble interaction held together by two hydrogen bonds with thymine or uracil. So it's a so-called T/UG wobble base pair. And then T, if you turn A round, flip it over, it forms a so-called Hoogsteen base pair, which is quite distinct. It's still held together by two hydrogen bonds, but it's different to the standard Watson-Crick interaction, which you see in duplex DNA. Now, what factors stabilize the duplexes? It's not all down to hydrogen bonding. You know, the bases themselves, they're aromatic structures with dipole moments. So there's a so-called base stacking interaction which takes place between successive base pairs. That's a complex mixture of hydrophobic van der Waals interactions and electrostatic, caused by where you've got, because remember, nitrogen and oxygen are more electronegative than carbon, so those bonds are polarized. But it's base stacking, basically, that drives the formation of the double helix. What it does is that the base pairs themselves are the least soluble, they're the least water soluble of the components that make up nucleic acids. They like to get out of water, so it's this base stacking that actually drives the ability of the molecule to fold, not to fold, but to sort of move and get out of water. And the polyanionic backbone, this is very, very interesting. You've got this repeating negative charge, which is Coulombic repulsion. So where you've got repeating like charges, remember, repel. Sort of something we learn at GCSE physics, or well, I learned it at O-level physics, but that shows how old I am. I'm mentioning that term now. But so what happens? This does something very interesting. This prevents the long sequence from folding up. Your electrostatic repulsion prevents that, so it tends to stretch out. And that allows base stacking and eventually Watson-Crick hydrogen bonding to occur. And this is very interesting because it's this repeating negative charge in the polyanionic backbone that actually it renders nucleic acids, it gives them, they're the one biopolymer in which the physicochemical properties are independent of the sequence. I mean, that's quite unique to nucleic acids. And what this does is this basically enables them to be sort of adequate purveyors of genetic information, and this enables Darwinian evolution to occur. In actual fact, you see when you've got G-rich sequences, you can see certain things happening, like a sort of tetramer complex forming, but by and large, the physicochemical properties of a stretch of DNA is independent of its sequence. So what are the constraints on a molecule which is carrying information, a genetic molecule? Well, it must be easy to replicate. In the modern world we live in, the modern sort of DNA, RNA, protein world, that is carried out by enzymes and cofactors, nucleoside triphosphates. But it was probably self-replicating in the precursor RNA world, which predated the modern biological world we live in. There's a lot of evidence for that, the RNA world. We see it all around us. I'm not going to go into that in detail in this talk, but I'm happy to answer questions about that afterwards. So, but essentially, a genetic molecule must be easy to replicate. And it must tolerate mutation. Any structural change, say if you swap an A for a G or whatever, or a T for a U, T for a C, that cannot compromise replication fidelity. So obviously you have things like mutations giving rise to different proteins that could either give the organism an advantage or a disadvantage, but in general I'm talking about any small changes in structure must not prevent the biopolymer from carrying out its ability to convey genetic information. That's essential. So what it should be capable of surviving modifications in constitution without loss of properties essential for replication. And that lends itself to a nice anagram, cosmic loper. You know, a genetic molecule must be a cosmic loper. Which is very nice if you're interested in thinking about life elsewhere in the universe, you know, it ties in quite nicely with that, I think. Okay. And this cosmic loper behavior is unique to nucleic acids. If you change the base sequence, that does not, in a stretch of DNA or an RNA, that does not change the physical properties or its chemical reactivity. You know, if you have, say you have two stretches of DNA, but 100 bases in length, you know, they'll go in an electric field, they'll migrate to the same place on a gel. It's because they're carrying the same number of negative charges. And so the physical and chemical properties are essentially independent of sequence. Now that's not the case for proteins. If you change an amino acid, one amino acid in a protein made up of maybe 20,000, that can have profound effects on that protein's chemical behavior, especially if it's an enzyme. And the best known one, of course, hemoglobin. That's it, hemoglobin. Changing the polar glutamic acid, which I've shown, the structure I've shown, with the non-polar valine at position 6 of the beta-1 chain, that leads to a form of hemoglobin that's much less soluble than the naturally occurring wild-type hemoglobin, and that gives rise to the disease known as sickle cell anemia. Likewise, here's a ribonuclease. This is a ribonuclease is an enzyme that chops up RNA. If you replace any of the essential amino acids in the active site, that's lysine 41, aspartic acid 121, histidine 12 or histidine 19, that will lead, if you replace any of those with another amino acid, with any other amino acid, that will lead to a loss of activity. So see, small changes in proteins or in enzymes profoundly affect their chemical behaviors and reactivities. Okay, this is the good thing about being a synthetic organic chemist. We can actually modify nucleic acid structures. We can make analogues of naturally occurring nucleic acids and test their behaviors in the laboratory. We can replace the polyanionic backbone, replace that sugar phosphate with something else. We can replace the sugars. Why has nature chosen deoxyribose or ribose? Why not glucose, for example? You know, glucose is actually the most abundant organic molecule on this planet. Why is it not used in our nucleic acids? We can replace the nucleobases with something similar, just a subtle juxtapositioning of the exocyclic amino and ketone oxygen groups, with just move those round, new heterocycles, new letters in a genetic alphabet. And of course, the big question is, how do these changes impact upon cosmic loper behavior? Will these change the ability, will that new polymer be able to act as a viable genetic molecule? So, we can but try. Now, if you take away the polyanionic backbone with its repeating negative charge, that has a profound effect. Now, for example, this structure, you can't see it, we've replaced the phosphate, which links 3' to 5', with dimethylene sulfone. Now, that's a polar group. You know, the sulfur atom is less electronegative than oxygen, so it's polar, but it's not charged. So I was working in a group, we actually did make, with a lot of sweat, blood and tears, we made oligosulfones and tested their behavior. We found something very, very interesting. They would not form duplexes to their complementary sequence. The reason for that, there's no negative charge here, so these analogues behave like proteins and kink and fold. So by replacing the polyanionic backbone with these dimethylene polar but uncharged dimethylene sulfone groups, we've created a nucleic acid that's more like a protein. So this could not be used as a genetic molecule, we don't think, anywhere. You're unlikely to find any alien life forms that use this as their information storage systems. I can happily conclude on that. And if you want to look at the article in Journal of the American Chemical Society back in 1996, I'll give any journal references I have, I'll happily send on to people who'd like to read them. There's one exception to this rule: peptide nucleic acids. This came out of the Panum Institute in Copenhagen at the same time as we were working on oligosulfones. A group led by Peter Nielsen in Copenhagen produced this molecule. It's an N-ethyl glycinal backbone in place of the sugar phosphate. But if you notice, it's got the same number of backbone bonds as naturally occurring DNA and RNA, and there's the same number of atoms between the backbone and the bases. You can compare as a direct comparison with naturally occurring DNA or RNA. This was Peter Nielsen, it won Molecule of the Year in Science in 1991. Now, these peptide nucleic acids form duplexes with themselves and with naturally occurring DNA and RNA. There's a model of the PNA duplex with a DNA duplex on the right. So, basically, could this be an alternative to naturally occurring DNA and RNA as a genetic information storage system? Well, the answer is no. As you get longer sequences, they become much less soluble. So when you go beyond about, I think, 30, they'll start to precipitate out of aqueous solution. Rather like the dregs in a beer barrel, that's what you'd find. So basically, become much and in fact become insoluble. So I don't believe these PNAs they're not cosmic lopers. You will not find anything in the universe which uses these. Now I say that guardedly, but just based on chemical logic. The question is, why phosphates? Why has nature chosen phosphates? Why not something else that can ionize at physiological pH around between 7 and 8? Well, the first pK of a phosphate ester is about 2. That means at pH 7.4, it's always ionized, so it's always charged. And this negative charge does something useful, especially in an aqueous solution. People tend to think of water as as basically almost inert. It's not. It's actually it's a reasonably good nucleophile. This negative charge prevents nucleophilic attack at phosphorus. So it gives a stability, a reasonable degree of stability to the nucleic acids. And also, if you look at the hard data, phosphate esters, that's both triesters and diesters, are much more stable than their carboxylic and other ester counterparts. And also, phosphate is geologically abundant. It's about 0.1% of the Earth's crust, about 10,000 parts per million. So it's abundant, so it makes sense that life would make use of materials that are relatively abundant. The great article, Why Nature Chose Phosphates, by the late Frank Westheimer, published in 1987 in Science. I have copies. I'm quite happy to send PDF copies of this to anyone that requests it. I strongly recommend you read it. It's brilliant. Here we are. Look at it. Let's compare the phosphate link with other potential links. This one, the structure on the left, that is citric acid. So, for example, if citric acid could be used as a linker between the nucleosides, remember also carboxylic acids, their pK is around, depending on the structure, it's around 3 to 4.5. So they're pretty much always ionized at physiological pH. The problem is if you look at the structure carefully, you'll find that one ionized carboxyl group doesn't shield the electrophilic centers very well from nucleophilic attack. Whereas the negative charge on phosphorus does indeed do that. That's a much better shield for the phosphorus atom than that ionized carboxyl is for the two electrophilic carbonyls of this citrate nucleic acid, if you like. Now, silicon, I think Sean mentioned silicon. Well, you can with some difficulty make a silicate-linked nucleotide. The problem is the OH group on silicon, the pK is 9.5, so it's a much, much weaker acid than phosphate and phosphoric acid. So that means at physiological pH, this would not be ionized. And moreover, that silicon atom is even more electrophilic than the carbon atoms in citric acid, which I've denoted. So this would basically fall apart very rapidly in aqueous solution, and in fact it does. You have to making these. You have to basically keep them away from water, keep them under nitrogen. They fall apart too rapidly. So you will not see citrate nucleic acids. You will certainly not see silicate nucleic acids. And here's the saponification rates of phosphate esters in comparison with, in this case, ethyl acetate. This is a rate of reaction in 1 mole of sodium hydroxide at 35 degrees centigrade. And you can see from the data that the phosphodiester is about 5 million times more stable than a carboxyl ester. So it's all down to the fact that it ionizes, and it's also much less prone to hydrolysis. And there's a mystery reported in Mono Lake a few years ago. It was in the news. It was all over the news. It made the popular news back in around December of 2011. Mono Lake, I knew where this place was. Does anyone else know? Maybe the surround sound might give you a clue. I first heard of it through this picture. This was a postcard inside a certain album released in 1975. That's the proper cover, and it's Wish You Were Here by Pink Floyd. To this day, I still play it. It's one of my favorite albums of all time. I used to enjoy when in the late 70s, I would watch the sun setting, put this on, watch the sun setting, and light up a large joss stick. But anyway, this mystery, it concerned, a lake mystery. You could think of a lake monster, but no, this concerned a microbe. And in actual fact, this picture was actually done for real. There was no Photoshop in those days. This poor chap had to sort of dive into this water I think it had with a snorkel on, and it's water that's highly alkaline, pH 10, and it's also very rich in arsenate salts. So I feel sorry for him. I wonder what state he was in when he came out, you know. What they put now, that would breach health and safety straight away. That just could not be done nowadays. Anyway, as I say, this microbe, this was isolated from the sediments of this lake, and a group of NASA scientists started growing these microbes in increasingly alkaline, arsenate-rich media. They actually claimed to be able to grow them in arsenate media only with no phosphate. And they went on to make the outrageous claim that these microbes were incorporating arsenate in place of phosphate in their essential biomolecules, including nucleic acids. So arsenate instead of phosphate. And this was published in Science the end of 2011. I have the paper if anyone's interested. But now this is indeed an extraordinary claim. And I'll have to leave the music there. We'll have to go on. We must go on. What a pity. I don't know if any of you know this man. Does anyone recognize this chap? Yes, I know. Yes, I know that Dan does. Well, he was a great popularizer of science, particularly the astronomical sciences, about 30 years ago. This is Carl Sagan. Sadly, no longer with us. He died at the very young age of 62 in 1996. His TV series Cosmos was one of the best ever science series produced. It came out and was released in America in 1980, the same time as David Attenborough did Life on Earth. I mean, they're two landmark science series, I do think, two of my favorite. Well, Carl was a great communicator. Among the people he inspired was a certain, at the time, an 11-year-old Lancastrian who's now all over our television screens. Yeah. Carl was also a great communicator. He had great quotes, but a quote that's very relevant to a claim made by Felissa Wolf-Simon and others concerning the microbes in Mono Lake. Extraordinary claims require extraordinary evidence. So let's have a look. Let's compare what we know about phosphate esters and arsenate esters and see, chemically, if their claims stand up. So phosphate v arsenate. Well, it's been well known that arsenate esters are much less stable than their phosphate counterparts. For example, in 1 molar sodium hydroxide at 100 degrees, the hydrolysis of dimethyl phosphate goes with a half-life of about 24 hours. So that means half of it will have, the concentrations of the two on the left and the right will be approximately the same after 24 hours. Now let's have a look at an arsenate counterpart, in this case diisopropyl arsenate. This in neutral water at 25 degrees, so just that water at room temperature, it's completely hydrolyzed within 2 minutes. And this is actually quite a good model for DNA linked together by arsenate. This is secondary, very like the 3' the 3' carbon in the nucleic acid sugars. So I mean the evidence is there. So it doesn't seem possible to me that essentially inside a microbial cell, which is essentially an aqueous solution, that they could have DNA constructed with arsenate instead of phosphate. And I think the evidence is strongly against it, and they haven't shown. They have all they did, as I said, was grow them in. They claimed a nutrient broth that contained no phosphate. Many have speculated that you can't completely, with arsenate, there's always going to be some phosphate. Yeah, you can't completely. How do you actually end up with completely pure arsenate? And also, if you look at compare the geological abundances, phosphate 10,000 parts per million, arsenate on average 5 parts per million in the Earth's crust. Now, Mono Lake, admittedly, its concentrations are much higher. What I think the NASA scientists were seeing was a microbe that was extraordinarily tolerant of high levels of arsenate. And it's probably incorporated them into vacuoles, which are then later expelled. That's what I think they're actually seeing. I mean, as I say, why would life make use of something that's very rare? You know. So now let's look at alternatives to the sugars. We've looked at the backbone. We've seen that essentially a phosphate, polyphosphate backbone is essential. Now, can we tinker with the sugars? Can we replace deoxyribose and ribose with something else and see what happens? Well, yes, here we are. This is a simple. This is an open. This is a glycinal. This is in glycerol DNA. You've got, this is like an open chain version of ribose or deoxyribose. It's the same number of backbone bonds and the same distance from the base to the backbone. But as you notice, it's not a member of sugar. These are 5-membered rings. In this case, you could have a lot of free rotation about these bonds, which you don't have in the naturally occurring DNA and RNA. So what do you think? I mean, I think, how would this behave? Well, let's have a look at how would we study. You make two complementary strands with this glycerol nucleic acid. How would it behave? Well, what you do is you measure the so-called melting temperature of complementary DNA. This provides us with a measure of duplex stability. And basically what this means, the melting temperature, it's the temperature at which half of the DNA is in the duplex form and half has dissociated into single-stranded forms. As you raise the temperature higher, more of the duplex dissociates into single-stranded forms. So the melting temperature is a measure of stability of a duplex. That's what we use. So we make a new nucleic acid derivative with either different backbone, different sugars, different bases. We study the melting temperature of two complementary strands. And for glycerol DNA, we replace you can replace 1, 2, or 3, the naturally occurring ones, with this open chain glycerol counterpart. Let's have a look at the duplex stability. The small t denotes glycerol linkages. But when you've got a small t, that means it's a thymine bound to a glycerol nucleoside. So that's a duplex. That's a naturally occurring duplex, melting temperature of 40. Now, what happens if we replace one of the sugars with the glycerol? Denoted small t, that means it's appended. You've got a thymine base appended to that glycerol unit. What does that do to the melting temperature? Well, it decreases it quite considerably. So you've gone from 40 degrees to 25 degrees. Now what happens when we put 2 of these open-chained glycerol units in? What happens? The melting temperature falls to really quite low, 13 degrees. And if we put 2 together in the sequence, the melting temperature falls to just 11 degrees. So clearly, this open-chained glycerol linkage would not be useful as a nucleic acid. Clearly not. You know, if they were, that's just with 2 linked together. That's with 2 separated by, in that case, 1 naturally occurring unit. So you can imagine two complementary strands. The melting temperature would be less than 0 degrees. And this work was published in 1990. So clearly, glycerol nucleic acids would not be cosmic lopers. Okay, why why five-membered ring sugars and not six-membered ring ones like glucose? I mentioned it's the most abundant of all the sugar molecules on this planet. And it's this great Swiss physicist, sort of physical chemist, physical and synthetic organic chemist, Albert Eschenmoser. This was his retirement project, by the way. In 1986, at the age of 65, he initiated a comprehensive study of nucleic acids containing six-membered ring sugars, so-called hexose nucleic acids. He synthesized oligos containing naturally occurring six-membered ring sugars, such as glucose and allose, and their dideoxy counterparts. The results were interesting and incredibly intriguing. So here we are. This is allose, maltose, mannose, and best known D-glucose. And some dideoxy ones, where these are just this is dideoxy allose. You've got 2', 3'. That's just allose without the 2' and 3' OH groups. And a pyranoside, this is like a six-membered ring version of ribose. Okay, so how do you think glucose? Why is glucose not found in nucleic acids? Yet it's the most abundant, basically the most abundant organic molecule on this planet, in the biosphere anyway. Well, basically you've got too many OH groups. They're larger, you get intra-strand steric clashes. As Eschenmoser himself put it, these sugars have too many atoms. As you can see, here's a glucose. This is glucose DNA. And you've got clashes where I'm putting in these arrows. You're seeing clashes between OH groups and the edges of the aromatic bases. And it was found that they don't form stable Watson-Crick bonding duplexes. So that's why glucose is not, it may be the most abundant sugar on this planet, but it's not used in nucleic acids. That's why. Now, modeling might have predicted that, but until you actually make it and test it, you can never know, because modeling can't predict everything with 100% fidelity. Now, and also, 2', the dideoxy analogues, they also display different base pairing to what we see in DNA and RNA, which is very interesting. This is 6', 4' homo DNA. You see these other Watson-Crick interactions, sort of Hugstein adenine AA and GG pairings, something you don't see in naturally occurring DNA and RNA. And in fact, if you look at this is 1, 2, 3, 4. These hexamer duplexes, it's a melting temperature of 34 degrees. It's displaying AA and GG pairings. That's comparable with the melting temperature of a naturally occurring hexamer duplex. And the pairing rules, while GC is still the strongest, that's still the strongest, but it's greater than, then followed by AA and GG. Both of these are stronger than the naturally occurring AT. So by changing these hexose sugars, you see some interesting affects the pairing rules. Now, pyranosyl RNA, that's a sort of six-membered analog of RNA, that forms extremely stable duplexes, so-called 4', 2' pRNA. These are more stable than naturally occurring DNA and RNA, and they've show exclusively Watson-Crick base pairing. So far to date, no non-Watson-Crick base pairing or Hoogsteen pairs have been seen for this particular nucleic acid analog. So that's very interesting. So that possibly could replace, you might come across aliens with this in their genetic information system, possibly, unless I'm not sure, I presume Hoogsteen pairing might provide have some biological efficacy. I've been looking it up. I can't see any of yet where it's been shown to be essential for certain biological processes. If it is, then this would not do. Okay. And finally, it was a breakthrough 3 years ago. It was in the lab of this chap. I don't know, can you see? Does he remind you of anybody, by the way? This is Philip Holliger, his laboratory in Cambridge. The clue I'll give you, he is Swiss, by the way. That usually then, people then usually get it. For me, he's a bit, well, I suppose it's that doesn't, the screen, you can't see it properly, but for me, he's rather like I would say, a cross between Roger Federer and an indie band singer from the 90s, I would say. But that's who he reminds me of. Apparently, he's in his younger days, he was tired of being told people think it was he Federer, apparently. Yep. But he's also worked in the group I once, we didn't work together at the same time. So he used these following pentose sugars. These are different to naturally occurring ribose and deoxyribose. The LNA, that's kind of locked RNA, that's got an ether linkage between on the 4 prime. It's got on the 4 from the 4 prime to the 2 prime oxygen. 3-O, this is TNA. That's if you compare that with a naturally occurring, it's a bit like the CH2OH group has been lifted from the 5 prime and appended to the 4 prime. Arabinose nucleic acid, that's ARA, also known as ARA-A. That's with the 2' OH in the opposite, in the different stereochemistry. And finally, fluoroarabinose, that's with that OH group replaced by a fluorine. He also looked at some hexoses. This is 1', 5' anhydrohexanol nucleic acid, or HNA for short, and a cyclohexenol nucleic acid, or CENA for short. What he's actually managed to do has been really intriguing and inspiring. These XNAs, as they're called, they form stable Watson-Crick duplexes to both themselves and to naturally occurring DNA and RNA. Like so. Nice duplexes. It's a HNA duplex. Moreover, what's intriguing, they'll actually do PCR. You can actually do PCR using these XNAs. This was a real breakthrough. The XNAs can be used as a template to make copies of them. They can amplify themselves. They can also amplify naturally occurring DNA or RNA complementary sequences as well. You can actually translate the sequence from XNA into naturally occurring DNA and vice versa. He had to develop some artificial polymerases that would accept them. But once he had those up and running, basically, the sky's been his limit. This is a real breakthrough. At last, these are sort of like synthetic nucleic acids that behave exactly like naturally occurring nucleic acids. Okay, now let's look at the base. So we've shown the phosphate is essential, but the sugars, we can be a bit more flexible. We can change them and still have something that behaves like a viable genetic polymer. What about the nucleobases? And this research was started in the group of this chap. This is Stephen Benner, who I had the pleasure of working with for many, many years. began research into an expanded genetic information system known as AGIS back in 1986. It's almost 30 years ago next year. He was intrigued by the question, why does life as we know it use just 4 nucleobases in DNA and RNA? Were additional ones used in the precursor RNA world? It's a good question. Might alien life utilize different or indeed additional nucleobases to the standard ones? And most importantly of all, when you're trying to seek funding from bodies made up largely of accountants, failed lawyers, and second-rate arts graduates, can this expanded genetic information system have technological or medicinal value? No. So non-standard nucleobases, if we look at, there's the AT pair, PY, and that means acceptor. This PY, that denotes pyrimidine, and the ADA is acceptor, donor, acceptor. It interacts with PU, DAD, with donor, acceptor, donor. And then, of course, we have the CG base pair. Remember, the CG base pair is linked together by 3 Watson-Crick hydrogen bonds. Now, what we can do is, if you look at the CG base pair, if we swap around the groups, the exocyclic amino and ketocarbonyl groups, this gives us iso-C and iso-G. They're analogous to C and G, just with the group swapped around. So instead of it's PYDAA, in iso-C, it's the opposite way, it's PYAAD. And the same for the purines. But that's not all. Other possibilities, xanthocene, that's the purine there, that does occur in nature, by the way, but not in nucleic acids. Then that will interact nicely with this pyrimidine analog. So by clever construction of heterocycles with amino groups and keto-oxygens, we can indeed construct more than just the 2 base pairs we see in nature. That pair is known as kappa and X, X for xanthocene. And there was 2 more using the same within this Watson-Crick possible in this Watson-Crick paradigm. That gives us a total of possible of 12 letters are possible in the Watson-Crick hydrogen bonding paradigm. That one is Z and P. This had a lot of success with this particular group, as you'll see in a minute. Now, this nucleobases, non-standard nucleobases actually entered popular culture. So if you remember E.T., came out in 1982. And remember the line, E.T. has DNA but with an extra base pair. The extra letters are inosine and a pyrimidine that we cannot identify. Well, why? Why? Clearly, Steven Spielberg didn't talk to Stephen Banner or anyone else for that matter. Because if you think about inosine, that's basically hydrolyzed adenine. It's adenine with the NH2 group removed. So it's got an acceptor donor. It's not hard to think of a pyrimidine that would form a Watson-Crick pair with that. That would be a possible complement. But why couldn't they see that? But then again, they're filmmakers. And also, remember this great series, The X-Files. There was the Erlenmeyer, which, by the way, they're making a new series. There's a new series in the making, apparently. But the best one, I think, was the Erlenmeyer Flask episode in 1996. A DNA sample from an alien body was found to have DNA with an extra base pair. Didn't say which one, but... Okay, so in fact, duplexes containing these non-standard bases are very stable. Do the melting temperature studies. For example, this is a 3, this 1, 2, 3, 4, 6. It's 10 more. We've got X and Y, and melting temperature. So when X and Y are C and G, melting temperature is 52 degrees. So now let's replace X and Y with some of these non-standard base pairs. Iso-C and Iso-G, it's also 52. It's no surprise, you just got the bonding patterns reversed, but they're still the same order of magnitude. Kappa and X, it's slightly higher, it's 58 degrees. And with the Z and P, it's 62. So, in fact, with Z and P, it's actually even more, it's 10 degrees, it's more stable. Duplexes containing the ZP pair would be more stable than those containing CG. And there's many similar examples in publication which show where several of them are present. So that basically the nucleic acids behave exactly the same. And we've also got here, rather, didn't do this particular piece of the work, but the Benner group, they've actually got DNA polymerases, both wild and synthetic, that basically construct NSBs and put them opposite their Watson-Crick partner with the same order of fidelity as they would the naturally occurring ones. And also, PCR systems have been developed that replicate the non-standard bases, with the same level of fidelity as they do the naturally occurring AT and CG pairs. So this is a real breakthrough. So this is similar to what Philip Holliger was doing with his sugars. Stephen Benner has done it with the bases. The best PCR systems, the ZP pair were better than Iso-C and Iso-G. That's been found. Okay. And I'd mentioned about technological use. In this case, medicinal value. There's a branched DNA quantiplex HIV and Hep A, B, and C diagnostics probe. It's used to monitor the health of patients on multiple antiviral drug regimens, in particular HIV and Hep B. Starts off with, in this case, a capture probe, which is complementary to sequences on a messenger RNA from either of these viruses. And now the trick is bringing in branched. There's a sort of the label extend. This part is complementary also to part of the viral messenger RNA. The solid lines contain just naturally occurring bases. The dashed lines contain non-standard base pairs. And so oligos with non-standard base pairs will only bind to their complementary sequence, but they will not bind very well to naturally occurring sort of DNA or RNA. And this makes this technique more sensitive than similar probes that use essentially naturally occurring DNA or RNA. You can see that that's the sequences. So the dashed lines contain Aegis, in this case Iso-C and Iso-G base pairs. Then there's a branched DNA also containing non-standard base pairs. So this is like a quaternary sandwich complex. And the final part of it, using probes, fluorescently labeled probes, also Aegis sequences containing fluorescently labeled probes. And these bind to their complements on the branched structure, like so. And it's a fluorescein labeled Iso-C, Iso-G. That's what the probe is. That's what the fluorescent probe is. And this can actually detect as few as 50 molecules of HIV or Hep B messenger RNA per mil. This is actually, as I said, this diagnostics probe is actually used it's still used to monitor the progress of patients on multiple drug regimens aimed at both HIV and Hep B. and some weird non-standard bases. This is the work of this chap, Floyd Romsberg. Now this is quite thinking outside the box. These are hydrophobic bases which don't interact by hydrogen bonding. It's something that I would not have believed, we would not have believed possible 25 to 30 years ago. And there they are. There's the two pairs. This is a D5,6, and its complement is DNAM. And there's DTPT3, and it will also complements with DNAM. Now these don't form hydrogen bond interactions. What they form is hydrophobic or stacking interactions. Like so. The duplexes containing these are stable. You can see they're forming like pi stacking interactions. Whereas you see the naturally occurring base pairs either side of them, they're like, these things are stacked on top of each other, whereas the naturally occurring ones are sort of like adjacent to each other to enable Watson-Crick hydrogen bonding to occur. That's not possible with these. And also he's gone further. He's got DNA polymerases that will make oligos containing these hydrophobic non-standard bases, and they'll put them opposite their complementary partner. They don't work quite as well, however, if you've got 3 or more in the strand, whereas in Stephen Benner's lab, those with iso-C, iso-G, or ZDP, they work equally as well with no matter how many you have. And he's also got PCR systems capable of replicating oligos containing these. But as I say, they don't work so well when 3 or more are in sequence. So you're limited to having them just a few. They have to be well spaced by naturally occurring bases. But there's something else. He's actually gone a step further. A bacterial plasmid containing a D5,6 DNAM base pair was constructed in 2013. It was taken up by E. coli, and the modified E. coli only replicates when fed these non-standard base triphosphates, in addition to the naturally occurring triphosphates, ATP, CTP, TTP, and GTP. So this is kind of like a non-standard organism, if you like, which has got one base pair, one D5,6 DNAM. It's the first organism that can grow and replicate with a most unusual unnatural base pair in its DNA. So Steve Benner's group has not tried this, but I'm sure he would there's no reason why he couldn't do the same experiment with either iso-C and iso-G or Z and P. Okay. Just a mention about the so-called antisense therapeutics. This was all the rage about 20 to 25 years ago. The idea being that if you could come up with an oligo that would interact with messenger RNA before it docks on the ribosome, you could prevent the information for a rogue protein, say for example a growth factor in a cancer cell, or for that matter a viral protein, from being synthesized. It was seen as a great way of shutting down protein synthesis, and hence a sort of huge leap in the area of medicine, so tackling especially certain incurable diseases like certain types of cancer and HIV at the time. And unfortunately, what happened, a lot of the drug companies invested hugely in constructing artificial DNA mostly with sort of like uncharged backbones, and pretty much all of them failed, except for the PNAs, all of them failed. And so the reason I'm mentioning this is to show you that a good Blue Skies project could help guide this. Like the groups of Stephen Benner, Albert Eschenmoser could have helped guide this antisense approach, rather than having them almost like competing side by side. That for me is the importance of doing fundamental blue skies research. You know, I remember when Michael Faraday was asked during one of his lectures when he was pushing a magnet in and out of a coil of wire which is connected to a galvanometer, the needle on the galvanometer moved. He was asked by a woman in the audience. She said, "Pray sir, what use is that?" His response was, "Madam, what use is a baby?" You know, and that kind of, for me, sums it up. The importance of doing fundamental research. And so the conclusions, the polyphosphate backbone, this is crucial for all genetic molecules. The sugars can vary to some degree, as we saw the work of Albert Eschenmoser and Philip Holliger. And other heterocyclic base letters are possible. We've seen the work from Stephen Benner and indeed Floyd Romsberg. So ET might not have DNA, but it will have a sugar phosphate in its nucleic acid. That I would safely predict. Yeah. And this work could be best described as one nanostep for a scientist, but a quantum leap in our understanding of nucleic acid structure. Just the acknowledgements. Of course, acknowledgements Stephen Benner for giving me the chance to live and work, join his group, first of all in Zurich in Switzerland and then in Gainesville, Florida. I was in the group for 6 years. I thank the Swiss National Science Foundation and NASTRA's astrobiology program for generous funding of this research. If there are any, and the US National Science Foundation as well towards the end. Thank you.